Hello, that's me. Yep. Wow, what a weird aspect ratio. Okay. So I thought I would try and find a title that was somewhat reminiscent of the title of the conference. But, of course, this is basically what SENS Foundation does, SENS Research Foundation as it's now known. We're in the middle of a rebranding exercise, so you'll see SENS Research Foundation here and there and SENS Foundation in other places. I apologise. We'll sort that out soon enough. And this is what we do, as many of you already know. We're interested in doing something about aging. We're not in favour of it. And we specifically hope that we will be able to bring it under extreme medical control using regenerative medicine. So, what is regenerative medicine? You've heard of stem cell therapy, you've heard a little bit today about tissue engineering and such like. But of course, regenerative medicine is broader than that. It includes everything that is to do with restoring the function of the body or the function of an organ by restoring its structure. And that could be at the level of replacing whole organs, or it could be at the level of replacing cells and thereby repairing organs, or it could be at the level of repairing cells by restoring the structure of the insides of cells, or even the material outside cells that holds the cells together. Aging also is a complicated thing to define, actually, a controversial thing to define. Some people define it in ways that are not really very useful for for orienting our thoughts around doing something about aging, around designing medical interventions. This is a really good definition that does orient our thoughts. It's mechanistic, it says how aging happens, that it's a side effect of being alive in the first place, which is what metabolism is. It also says that the way that metabolism, being alive in the first place, causes the diseases of old age is by causing the gradual, lifelong accumulation of various types of damage, various types of microscopic, molecular and cellular changes to the composition of the body and the structure of the body, which the body can tolerate for a long time, because it's set up to tolerate a certain amount of damage, but which eventually overwhelm the body so that you get the diseases and disabilities of old age and eventually die. You know, we've got a whole bunch of ways to think about aging, to combat aging, built into us. And a lot of the things that we see changing during aging look as though they may be, things that we could easily change back. Changes in how genes are being expressed. Some genes may make more protein in old age than in younger ages. Some genes may make less. But often it's actually a bad idea to try to change those things back, because many of these things change on purpose, so to speak, as an adaptation in order to minimize the rate of aging. And therefore, if we tinker with those things, then we actually end up being in a worse way than we otherwise would be. We're actually doing more harm than good. However, what that actually means is that when we want to combat aging, the number of things we have to fix is relatively small, because it's essentially restricted to non-genetic changes, changes that are not coordinated and therefore are probably not adaptive, not trying to minimize aging. So this is really what it all comes down to. Here's what I said about the definition of aging earlier on. Metabolism causes damage throughout life, which eventually causes the diseases and disabilities. And there are two ways that people have tried to deal with that problem over the years, tried to think about dealing with it anyway. One of them is what I call the geriatric approach, which is essentially patching up, papering over the cracks of aging. It's trying to attack the diseases of old age as if they were normal diseases, like infections, and eliminate them from the body. And that's obviously ridiculous if you think about it, because how can you possibly eliminate from the body something that's being caused as a side effect of the body's normal operation? It's obviously impossible. So the geriatric approach is just never going to work. It's a little better than nothing, but it will never be much better than nothing. A lot of people have realized that before me, and they have been working on trying to essentially adopt the policy of prevention being better than cure. So they try to intervene at this earlier stage in the chain of events and clean up metabolism. The idea here is that if we can slow down the rate at which metabolism creates damage, then we've got a much better chance of actually postponing the age at which these pathologies emerge. But it turns out that that's really not a good plan either, for a very different reason. The reason that's not such a good plan is here, metabolism is really rather complicated. And, you know, if you try to tinker with this in a manner that significantly reduces the rate at which it does what you don't want it to do, namely create damage, then you're almost certainly going to, you know, to have side effects, to do other things that you didn't expect that do more harm than good. Actually, this is an understatement of the problem. This here is a simplified diagram of a small subset of what we know about how metabolism works. And even that understates the problem, because what really matters is that this is a simplified diagram of a small subset of what we know about how metabolism works, which, as any biologist will tell you, is completely dwarfed by the astronomical amount that we don't know, even ignoring all the stuff that we don't even know that we don't know. So it's hopeless. But luckily, the whole situation is rescued by the fact that there is a third approach that people have not really looked at until the past decade or so, since I've been promoting it. And I'm going to call it the maintenance approach. You can think of it as the regenerative medicine approach, repairing the damage that's occurred. Here what we're trying to do is we don't try to attack the process where damage creates pathology. We don't try to attack this process where metabolism creates damage. What we do instead is we separate those processes from each other by periodically going in and repairing this damage so that even though it's being created, it doesn't actually reach this level that's problematic to our health. And I claim that this is likely to be a much more practical approach, essentially because it avoids the problems that I outlined earlier with the other two approaches. The problem of being too late in the game and just patching up something, and the problem of trying to interfere with something we don't understand. It gets better. Not only is it like conceptually the right place to intervene in this process of aging, the weak link, so to speak, it also turns out that the number of things we have to fix is relatively manageable. Or at least, the number of types of thing we have to fix is relatively manageable. So, I pretty quickly, when I realised that this might be a promising way to go, I pretty quickly realised that there were really only 7 different types of damage that we need to worry about, and they're listed here. As you can see, these things are proper concrete biological phenomena, things like loss of cells, that just means cells dying and not being automatically replaced by the division and differentiation of other cells. Things like cells having too many cells because they're dividing when they're not supposed to. Having too many cells because they're not dying when they are supposed to. That turns out to be important in the immune system. We can go down this list and look at all these other ones. These are, the other 4 are not so much cell number, but rather molecular. 2 of them are what happens inside the cell, these 2, and 2 of them outside the cell, these 2. And they are, again, things that are caused as side effects of normal metabolic processes, but they are accumulating because they are not repaired automatically by the body as they're created. So they all, and this one here, mitochondrial mutation, is a bit of an exception in that we don't really know yet why it's bad for us. But there's lots of circumstantial evidence that says it is bad for us, and therefore that we ought to fix it, though the manner by which it's bad for us is probably rather subtle and elaborate. But all of these other ones, we can point to particular types of age-related disease which are mainly caused by these things. So cell loss, for example, we can take Parkinson's disease. That's mainly caused by death and non-replacement of a particular type of cell in the brain. Cancer, of course, I mentioned the immune system, the aging of the immune system. Molecular garbage inside the cell. This is actually the reason why we get cardiovascular disease and also macular degeneration. I'm going to say a bit more about those things in a few minutes. Molecular garbage outside the cell may be important in Alzheimer's disease. We certainly see a lot of it in Alzheimer's disease, though it's still controversial whether that's actually a part of how the disease progresses. But it's much more unambiguously the cause of something called senile cardiac amyloidosis, which turns out to be the #1 killer of extremely old people, people who get up to the age of 105 or 110. So that's definitely something we have to think about as well. And finally, at the bottom here, the cross-linking of this lattice of proteins called the extracellular matrix, which holds our cells together. turns out to be the reason why we get hypertension in old age, which of course has loads of effects I'm sure you're familiar with, things like exacerbating diabetes and kidney failure and such like. So we can point to very clear pathologies that doctors know all about and look at their early stage etiology, the origins of those diseases, and we see that they are side effects of the way that the body normally works, and that they accumulate throughout life. They're initially harmless, like I said, harmless for a very long time, until the body gets to be overwhelmed by the sheer abundance of these types of damage. Now, of course, a big question comes up immediately. How can I be so sure that this really is an exhaustive classification of the things that we need to fix? And I think there are a couple of reasons why we can say that. First of all, it's been the same list for 30 years. Since way before I came along, all of these things have been under extensive study by people who are interested in the biology of aging. And you really would think we would have added to this list by now if there were an 8th thing or a 9th thing to be doing. Plus also, I mean, I am the first person who actually spelt out in this rather, combative way that there are only these 7 things we need to fix. And a lot of people haven't really enjoyed hearing that. They felt that this was oversimplistic, but no one's actually come up with anything to add to this list in any convincing way for the past 10 years. So this classification does seem to be standing the test of time, and that's pretty good news. However, let me emphasize that we are not complacent about this. We are always on the lookout for possible things that might need to be fixed, in addition to the ones I've listed up there. And in fact, one of the projects that we're funding at the Aubrey College of Medicine in New York is looking at the only one that's really out there right now where there's a bit of a case where it might be important. So we're spending good money to make sure that if we are, make sure that we're not being overconfident in the nature of the problem. However, that's all very well, describing what the problem is and cutting it down into these 7 sub-problems. The question, however, comes, how can we actually fix those 7 sub-problems? And luckily we can answer that question too. So on the left here you see the same list that you saw a moment ago. On the right we've got the things that we are pursuing to try to address all of these problems. Now, we're not doing much in the top one. The reason we're not is because lots of other people are doing it already. You all heard of stem cell therapy, and if you think about it, that's exactly what stem cell therapy is. It's therapy to replace cells that the body has lost and is not automatically replacing on its own. I mentioned Parkinson's disease a few minutes ago as a classic example of a part of aging that's caused by cell loss. And Parkinson's disease is actually the subject of clinical trials already, not even work in mice anymore, but actually in human beings, to do stem cell therapy to actually cure the disease. At the moment, those trials are somewhat at an early stage. Certainly that therapy doesn't work every time by any means yet, but when it does work, it works spectacularly. There are some people who have been completely cured for a long time of Parkinson's disease by cell therapy. And so the general belief is that it's simply a matter of tuning the nature of the cells that you introduce so that they do exactly the right thing when they're in there. So that's really good news. I'm not going to talk much, unless people want me to, about what we're doing about cancer, because the therapy in question is very elaborate and complex and ambitious. It's actually by far the most ambitious part of this 7-point plan, and it would take quite a long time for me to explain in detail, but as I say, I'm happy to answer questions about it if anyone's interested. This business about death-resistant cells is fascinating. There have been a number of interesting studies recently about that, and I've actually got a slide or 2 about that later on, so I'm going to leave that to one side for a moment. Mitochondrial mutations, the mitochondria is this weird part of the cell that has its own DNA. It's the only part of the cell that has its own DNA. But it's an absolutely essential part of the cell. It's the place where the chemistry of breathing happens, where oxygen is chemically combined with nutrients to extract energy from nutrients. And the 13 proteins that are encoded in the mitochondrial DNA, they're needed. And when mitochondrial DNA becomes mutant, those proteins go missing, and we're trying to put them into the nucleus. We're trying to put the genes for those proteins into the nuclear DNA, modified in such a way that they still work. That's what's called allotopic expression. And again, this is not something that I introduced myself. This is an idea that's been around since the mid-80s, And it's only in the past few years that there's been real breakthroughs in showing how we might actually achieve it. So that's a big project in our research center in Mountain View, California. I'm going to talk a fair amount about this one, intracellular junk, later on, the molecular garbage inside the cell. These other two at the bottom, the two that are outside the cell, are also the topic of extensive work that we are supporting at universities around the world. In the first case, collaboration between the University of Texas in Houston and Harvard University, and the second one, a collaboration between Yale and Cambridge. So we're working with the very best institutions in the world in all of these areas, and that again is something that demonstrates how this whole approach to combating aging is really gaining credibility and legitimacy, and how the derision that some not terribly careful people heaped on it a decade ago has definitely disappeared into history. So that's all pretty good news. We have a fair idea how to fix all of these things. And if anyone's thinking, where's the detail? Do we really know enough about how to do all of these things that we can actually be confident of success? Well, of course, you can never be confident of success in research. We know that we may end up hitting obstacles that we didn't expect. But the fact is, we have a lot of detail about how we might go about all of this, to the point that when I wrote a book on all of this about 5 years ago, I was able to include an entire chapter on each of these 7 things. That's a lot of detail on not only the nature of the problem, but where we are in developing these solutions. Right, so I said I was going to talk a little bit about a couple of these things in more detail. First one, this one, death-resistant cells. So I mentioned the immune system earlier as a part of the body that goes wrong as a result of the accumulation of this type of damage. But it's not just the immune system. We also accumulate cells that get into a weird state where they can't divide when they're supposed to, and also they secrete proteins that may be carcinogenic, may induce unwanted proliferation of cells nearby. And so we really want to get rid of those cells. And just 6 or 8 months ago, no, about a year ago now actually, a group in Rochester, Minnesota, at the Mayo Clinic, published a very fascinating paper. Here are a couple of figures from it. Essentially what they did was they developed mice that they could artificially manipulate with regard to the number of these death-resistant cells. First of all, the mice had a mutation that caused them to accumulate these cells at an accelerated rate relative to what happens in normal mice. And secondly, they had a mutation, well not exactly a mutation, an engineered gene, that allowed the death-resistant cells to be eliminated when the mice were given a simple drug. So the idea is basically you create this thing called a suicide gene, which is normally latent, just sitting there and not being transcribed and translated into protein, but when the drug is introduced, it turns on that gene specifically in cells that are already making protein from some other gene. So the other gene in question in this case was this thing P16, which turns out to be overexpressed in these death-resistant cells. So they were able to kill the P16 overexpressing cells on demand at any chosen point in the lifespan of these mice. So here's a mouse that's about 10 months old, and it's not got long to live. That's less than half as long as a typical mouse lives, and that's because of this accelerated accumulation of these death-resistant cells. And here's a mouse that's the same age, and it's got that mutation as well, but at 5 or 6 months of age, it was given the drug to eliminate all the death-resistant cells. And lo and behold, it's obviously a much happier mouse than the one down here. And there's a million different, or at least a dozen different measures of biological age that were included in this paper. I've just put one up here for illustration. This is to do with muscle loss. So it was a really dramatic result, in a way. But here's the interesting thing. These people actually, it was a really pretty early stage proof of concept study in a lot of ways. First of all, obviously it was only in mice, so that doesn't necessarily translate to humans. Secondly, it wasn't a case of making mice age more slowly than normal, than normal mice do. It was a case of restoring normal rates of aging in mice that had previously been given an accelerated rate of aging. And all biologists know that actually it's typically much easier to restore something that you've broken than it is to enhance something that you haven't broken. The third thing that's interesting about this is that because of this trick about the suicide gene, the study didn't give us any information about how to develop a drug that could achieve the same thing in genetically unmodified mice, or humans for that matter. So again, it's a very preliminary study. It doesn't really tell us how to move forward. And the fourth thing, which is sort of the headline for sort of non-biologists, is it didn't even extend the lifespan. These mice here that had the cells removed, they still died at like 11 or 12 months of age, the same as these ones, because it turns out they had another problem in their heart, which was not caused by these P16 positive cells. It was some other bad effect of the mutation that caused the accelerated accumulation of these. So in loads of different ways, this was a study that did not really imply too much breathlessness. You know, it didn't really justify getting too excited, except from a scientific point of view. But yet, here's the interesting thing. Literally like weeks, less than weeks actually, like almost days after this paper came out, the people who were the main authors on this study founded a company and got a serious amount of seed capital from some venture capital fund or other, to actually go forward and develop a drug that would actually eliminate these cells from human beings. Now, that's pretty exciting. Here's why it's exciting. Aging has not historically been very fashionable as an area for venture capitalists to get into, to fund startups. There are, of course, a few notable exceptions, but there's very few these days. And I think that that's basically because people have come to believe that it's going to be a very, very long time before we can do anything significant about aging, and therefore they want to make money faster. They want a faster exit strategy. But this seems to be an indication that the wind may be changing there. That there may be now an increasing understanding that not only are we getting closer to developing these therapies, but also an understanding of the enormous magnitude of the market that will exist when the therapies come along, such that people who are thinking about investing in startups in the biomedical industry are now beginning to do different calculations in terms of the risk they're willing to take and the time frames they're willing to operate within, in order to get to the point of funding something like this. I'm pretty happy about this. I think it could be really a sign of things to come in terms of the private sector involvement in all of this area. Okay, so now I'm going to talk about molecular garbage inside the cell, intracellular junk. I'm going to talk now about some work that we at SENS Foundation are doing. The work I just described was not funded by us. This work is very much funded by us. It's work that we're very proud of. We've been working on it for 5 or 6 years now, and it's really finally coming together. So what I've got here is a picture of the very early stage of cardiovascular disease, atherosclerosis. In atherosclerosis, as you know, we get the accumulation of fatty deposits in the major arteries, and those deposits form these sort of globular things, these plaques, which sometimes burst. And the inside of the plaque, this fatty stuff, gets out into the circulation, and globs of it may get stuck in blood vessels in the heart, causing a heart attack, or in the brain, causing a stroke. And of course, cardiovascular disease is the #1 killer in the whole Western world. So how does that process begin? How does fat actually start to accumulate in the artery? Well, it turns out that the way it happens is as a regular side effect of normal metabolism. Cholesterol, normal regular cholesterol, is an essential molecule that cells need, and it's, of course, ingested in a diet, and it's also synthesized in the liver. And from those two places, it is transported around the body to other places. And it's transported in little particles called low-density lipoproteins. Those particles, it turns out, sometimes get stuck in the artery wall, especially the walls of our major arteries. And there's a type of white blood cell called a macrophage, which goes into the artery wall and engulfs this stuff, basically just swallows up bits of detritus, mostly this low-density lipoprotein stuff, and processes it, and just basically recycles it back into the circulation in a harmless way. Unfortunately, this process is not perfect, because these particles that have cholesterol in them, that are transporting cholesterol around, are also somewhat contaminated with material that macrophages can't process properly. This contamination is usually an oxidized version of cholesterol. I might do that in a minute, yeah. Okay. You know what, maybe it's just too complicated. Maybe I'll show you at the end. Some of you saw it yesterday anyway, at the event that we had. So this stuff that's accumulating in the artery wall is engulfed by macrophages, but the macrophages don't like it. And eventually they just get poisoned by it. They stop working, and they become this sort of undead inflammatory thing called a foam cell. A foam cell is basically a very sick macrophage. And these carry on accumulating in the artery, and eventually they get to the point where they start putting out signals and bringing in other macrophages that get into the same sort of trouble, because they also can't handle this oxidized cholesterol. So a long time ago I suggested a way of doing something about this. And essentially it came from the observation that this sort of oxidized cholesterol that we can't handle, that accumulates in the body during life, doesn't seem to accumulate in the environment. It just doesn't seem to be there. You'd think that, environments that are enriched in human remains would be enriched in the stuff that we're made of when we die, unless that stuff can be broken down by something in the environment. And of course, it does accumulate bones and teeth, but not oxidized cholesterol. So I reasoned that maybe this means that there are bacteria and such like in the circulation that, in the soil, that can actually get rid of this material. And maybe we could find out which bacteria, and we could find out what genes and enzymes they're using to break this stuff down, and then we could put those genes into our own cells, and that could be pretty therapeutic. Essentially, this is the suggestion. You've got this process in the body that turns young people into old people, and eventually into dead people. And then you've got this other process, a completely different process, that turns dead people into decomposed people. And that process is encoded in the microbial ecology of the environment. So we figure out what's going on there. We do standard molecular biology and cell biology. And we apply that knowledge to our own cells. This is actually supposed to be a neuron. It's a rather horrible cartoon. And that counteracts the initial process that turned young people into old people in the first place. All right, so the question is, well, you can clap when you've seen the data. The question is, can we do this? So we started out, as I say, a few years ago now, and it took us a while to get going, but after a year or two we were able to get results like this. This is actually from the first paper that was published from work funded by SENS Research Foundation, or the Methuselah Foundation as it was then. What you've got here is what's called an enrichment culture, where you take a whole bunch of different strains of bacterium and you give them the stuff that you'd like to get broken down. In this case, this one particular compound, 7-ketocholesterol, which is one of several different types of oxidized cholesterol that macrophages don't like, but it happens to be the most important, it's the most abundant and the most toxic. And 5 of these bacteria can't do bugger all with it, and they just sit there. They don't grow, and the 7-ketocholesterol doesn't go away. But these other 2 strains have a whale of a time. They've got genes that allow them to break down the material, and sure enough, after only 10 days, it's completely gone. Lovely. So that's step 1. Then we've got to find the genes that they use. It turns out that's not very hard either. We had a couple of techniques that we used for that. So then what? Well, we've got to figure out how to actually get those genes working in human cells. And of course we start out by trying to do that in cell culture. And that took us a while, because the fact is, the bacterium and a human cell are very different things. The bacterium doesn't even have a nucleus, or a lysosome for that matter. So we had to do a bunch of messing about and adaptation and modification of these genes to get them to work. But we now have. This was published a few months ago by the group that we're supporting at Rice University in Houston. And essentially what you're seeing here is that if you put in a completely humongous amount of this horrible substance, 7-ketocholesterol, then the cells are completely blitzed, whether or not they've got this bacterial gene that we discovered. But if you put more modest amounts in like this, then you've got a much taller bar there than the other bars here. So what that's telling you is that there's more survival, more viability of the cells that are given the compound than there is for these various negative control lines, where, for example, there's no enzyme, or there is the enzyme but it's being targeted to the wrong part of the cell, or it's the wrong enzyme, or something like that. So this is really very, very good news, very encouraging, and we're very happy about it. I think we're probably only a year, 18 months away from moving on to doing the same thing in mouse models of cardiovascular disease. Yes? Answer the question. So these cells actually are just fibroblasts. Actually right now, one of the first things that we want to do before we move to live mouse models is to do the same thing in other cell types, especially in macrophages, and that work is actually ongoing right now. We're pretty confident that it will work. Yeah. No, this has nothing to do with mitochondria. This is straightforward, putting these genes into the nucleus and having them expressed. Remember, the problem here is not mitochondrial mutations. The problem here is that the material is accumulating in the lysosome, and so we just have to target the enzymes to the lysosome, which actually turns out to be a good deal easier than targeting an enzyme or a protein to the mitochondria. Okay, so yeah, that's where we are with cardiovascular disease. But that's not the only thing we're interested in when it comes to molecular garbage inside the cell. This is another problem that's caused by exactly the same process, though in this case the molecular nature of the garbage is different. This is macular degeneration. Here's the eye, here's the retina, and the retina accumulates this sort of fluorescent stuff during life, and eventually that fluorescent stuff gets abundant enough that it kills the cells at the back of the retina, things called retinal pigmented epithelial cells, and that's why macular degeneration happens. That's the major cause of blindness, the number one cause of blindness in the elderly. We'd like to fix that, and we want to go about it in exactly the same way. The molecule that is mainly responsible for this, the molecule that accumulates to large abundance in the back of the eye, is this thing, A2E. It's got nothing to do with cholesterol. It's formed by a chemical reaction between vitamin A, which as you probably know is essential for the process of seeing in the first place, for how vision works, and fatty acids that are part of the cell membrane. But when it's created, it's a bit of a problem because the body doesn't know how to break it down. So we have done the same thing. We have been looking for bacteria and other species that can break this stuff down and finding out how they do it. And we found an enzyme, we're going to call it A2Ease. I'll show you a bit about that in a moment. Here what I'm showing is the experiments that we're doing to test for the ability of enzymes to break down this substance. So on the top row, all you've got is something showing where the lysosomes are in cells. This blue blob is the nucleus of a cell, so these are all different cells, and the red here is a stain for a protein that is always in the lysosome, so that's where the lysosomes are. This stuff, A2E, we then put into the culture media, just outside the cell. But we do it in such a way that the cells have a tendency to engulf it. They just pull it in, very much in the same way that I was explaining earlier, that macrophages in the artery engulf this detritus in the artery wall. So they pull it in, and they put it in the lysosome. We can tell that because of the overlap that we see here. This is this lysosomal stain again, this is the stain for our toxic substance, and here is this yellow which shows the overlap of the two. So that's good. We can get this stuff into the right part of the cell, in cell culture, without having to wait for the cells to actually create it internally the way they do in the back of the eye naturally. So here's the action of this experiment. Here's the enzyme we're talking about. I'm going to call it A2Ease. I'm going to call it A2Ease. And at the top here, we don't have any of that enzyme. So you've got your A2E, this is the stain for the A2E, there's no stain for the enzyme. And here's your, looks just the same as on the top left. But on the bottom, we've put in some enzyme. And first thing you can see is, even though we put in just as much A2E as before, we can't see any. it seems to have almost all gone away. Just a few little blobs. See a few little blobs here and there? Here is the stain for the enzyme, and here is the merge, the overlay. You can see those little blobs of remaining A2E are not yellow here, they're still green, which means they just happen to be lysosomes that the enzyme hasn't yet got to. So this is really excellent news. It's not quite so persuasive and definitive as the point that we're at with regard to oxidized cholesterol, but it's still very good news. We're pretty happy that we are, again, only maybe a year or no more than 2 years away from doing real studies in live mouse models of macular degeneration. So that's where we are with one of our major projects. It's still going to be a while before we can cure these diseases in human beings. I've told you all about how we've been able to do steps 1 through 4 on this thing. We've identified bacteria that do certain things, we've identified how they do it, we've made their enzymes modified so that they work in mammalian cells, and of course that they're not toxic. Getting onto mice is a big job. Even any mouse project takes a long time, so it's going to be, quite a few years before we get to clinical trials. But that's fine, I mean, someone's got to work on these really hard problems or we'll never solve these diseases. So that's really why SENS Foundation exists. I'm going to actually wind up now and give a bit of time for anyone to ask questions, because I know that this is a very mixed audience and I don't really know what sorts of questions people are going to want to ask. But I did want to mention this book that I wrote 5 years ago. I mentioned it already a little bit. I think it's still pretty useful, even though it's 5 years old. The good news is that there has been plenty of progress, some of which I've outlined for you today, in taking forward the various research programs that are described in this book. But the good news is that that has been largely successful, so that we haven't had to radically revise any of our plans and proposals, which means that the book, even though it's 5 years old, is not really out of date. And I'll stop there. Thanks very much. Okay, so before I take questions, I'm going to show you a couple of animations that we had made very recently.
They are new and they're really cool, and I think you should see them. Just played it for the first time last night.
Yeah, these are going to be part of our new website, which will be launched in the next few days. And they've been made by an absolutely stellar animation firm named Cosmosite, who work with Ted Mayer and other really prestigious groups. And the music, if I'm not mistaken, was composed by the lady to my left. So, it's indeed fantastic, as she says. I have no idea why this isn't working, but I'm sure that somebody will tell me. Okay, well done. Right, this is our intro.
We need cholesterol. All cells require it to function. And it's delivered to our cells via specialized particles in our blood. But sometimes these particles become trapped in the lining of our blood vessels.
Does anyone recognize the voice? Okay, well, the voice is Edward James Olmos, as in Captain Ajama from who is just the first of many celebrity supporters of SENS Foundation and of this work. And he's been very graciously giving us his time to do things like this for us.
White blood cells, called macrophages, clear their stuck particles by absorbing them and processing them in compartments called lysosomes. Here, the particles are broken down into their constituent parts for reclamation or disposal. Once the enzymes have done their work, the reclaimed cholesterol is returned to the blood. The problem is, some cholesterol becomes oxidized during its travels. Oxidized cholesterol is poorly processed by the macrophages and eventually overwhelms them, preventing them from even processing the normal cholesterol they absorb. These sick, bloated cells are called foam cells, and they attract other macrophages, but the newcomers suffer the same fate. As foam cells accumulate, they form arterial plaques, which become inflamed and burst, spewing out blood clots that trigger heart attacks and strokes. But what if we gave the macrophages a new enzyme so they could process oxidized cholesterol too? Then they would process stuck particles without getting poisoned and bloated and return to healthy function. This would progressively cleanse arteries of plaque that had already formed.
There you go. So it's all very well listening to me whitter on for 10 minutes in front of a microphone about all this, but I think when you can see it in an animation like this, it's probably going to be a good deal easier to get across to the general public. So we're very happy about this stuff. Yeah, so now the other animation, is a bit shorter, is simply a depiction of the early part of what I told you about how aging works with metabolism and pathology, and why the maintenance approach seems to be so promising. And again, you'll see that a picture is worth 1,000 words. We're going to be having several more of those made because we feel they are an enormous addition to our arsenal, our outreach arsenal. Maria, as you mentioned earlier, is one of the key people in our outreach team that we're putting a lot more resources into over the past year and going forward. And we hope that that's going to make a real difference to actually educating the public and bringing money in the door so that we can do this work a bit faster.
Basically, as you can see, we have a lot of this stuff figured out. We know a lot about what we have to do to achieve this mission, which is the most important thing that we can ever achieve, really. Curing the diseases of aging, curing the diseases of the world, preventing a lot of suffering and unnecessary dying. And we basically need money to do this. We need the right funding to advance faster. So if you are very wealthy, or if you know someone who is very wealthy, let them know and stay in touch. Come talk to me. We have a booth here. Please help us. Join us. Thank you.